fertilization. The process by which two gametes or reproductive cells, sperm and ova, having a single haploid set of chromosomes, fuse to become a zygote which develops into a new organism is called as fertilization. In case of some animals, the eggs develop into a new individual without undergoing fertilization. This phenomenon is called as parthenogenesis. Fertilization process is a specific for each species and not any sperm can fertilize any ova. But a specific lock and key type of a mechanism exists in which the ova produce fertilizin, which stimulates the specific sperm which releases the anti-fertilizin. This type of reaction is similar to antigen antibody reaction and helps in maintaining the species. Now, coming to the types of fertilization, there are two types of fertilization. They are external fertilization and internal fertilization. External fertilization, in this type of fertilization, both male and female gametes are released into the external environment, usually aquatic medium, where both the gametes fuse and fertilization takes place. Commonly, it is seen in case of invertebrates. External fertilization is seen in case of oviparous individuals, that is, egg-laying individuals. Internal fertilization, in this type of fertilization, one of the gametes is stationary while the other is motile. The stationary gamete is the female gamete and the motile one being the male gamete. Fusion of the gametes usually takes place in the female body as the ova are stationary while the sperms swim through the fluid medium and reach the ova. Now let us see the mechanism of fertilization. The process of fertilization takes place in 5 major steps. The following are the steps involved in the process of fertilization: mobility of the sperm towards the ova, capacitation and contact, acrosome reaction for penetration of sperm into the ova, and activation of the ovum. Finally, the fusion of the male and the female pronuclei, which is called as the amphimixis. First step involves mobility of the sperm towards the ova. Sperms are produced by the male individuals and ova by the females, as already we know it. So both the gametes have to come into contact with each other first for the fertilization process to take place. There are several ways in which the gametes meet. The initiation process is mostly chemotactic. The sperms swim towards ova and collide with them. Numerous sperms come in contact with the ova. In case of external fertilization, both the gametes are released in large numbers so that there are fair chances of the sperm meeting the ova. While in those animals with internal fertilization, since the ova is stationary, the male gametes are released into the genital tract of the female, where they move upwards and reach the ova. Now, then next step will be the capacitation and contact. The changes that occur in the male gametes, that is spermatozoa, which makes them capable of fertilizing the female gamete, that is the ova, is called as capacitation. The capacitation is related to the very fact that the ova of the same species is fertilized by the sperm of the same species. This is a chemotactic process which involves fertilizin and anti-fertilizin reaction, and this allows only few sperms to approach the ova. Acrosome reaction for the penetration of the sperm into ova is the next step in the mechanism of fertilization. For the process of oogenesis to be activated, the penetration of the sperm into ova is essential. For the entry of the sperm into ova, the acrosome of the sperm produces sperm lysins, which are the enzymes having the ability to dissolve the egg membrane. By lysing the egg membrane, the sperm enters into the egg cytoplasm. In case of eggs with thicker covering, the sperm enters through a special canal, the micropyle, or through receptor cone, which is an outgrowth in the egg membrane. In case of mammals, the eggs are encased in a layer of follicular cells called as corona radiata, which acts as a barrier through which the spermatozoa have to penetrate to reach the egg plasma membrane. The cells are held together by adhesive cementing substance, which is a mucopolysaccharide called as hyaluronic acid. The sperm's acrosome produces hyaluronidase, an enzyme helpful in dissolving adhesive substance which disperses the cortical cells. The acrosome forms into acrosomal filaments by undergoing morphological changes which help in the penetration process. The head and middle piece of the sperm only enter the egg, while the tail of the sperm is left outside the ova. Then the activation of the ovum forms the next step in the process of fertilization, so for which activation of ovum is the most important aspect in which the egg is activated to develop as it is in the inactive state. With the penetration of the sperm, very important changes take place in the cytoplasm of the egg. With penetration of the sperm, the plasma membrane of both the gametes fuse and a single continuous mosaic membrane is formed. The changes that occur in the cytoplasm of the egg are formation of fertilization cone, cortical reactions and fertilization membrane formation, and metabolic activation of ovum. For the formation of fertilization cone, as the contact of acrosomal filament of spermatozoa with the surface of the ova takes place, the cytoplasm bulges at the point of contact. The bulge is produced into a process of hyaline cytoplasm, which is called as cone of fertilization. The cone of fertilization gradually engulfs the spermatozoa. The physicochemical reactions that are set in the cortex of the egg due to the entry of spermatozoa into the ova are called as cortical reactions. These cortical reactions vary from one species to other. Cortical reactions usually involve exocytosis of cortical granules, the secretory vesicles, which are just below the egg's cytoplasm soon after the fusion of sperm with the ova. The cortical reactions lead to the formation of fertilization membrane outside of the egg's plasma membrane. This membrane inhibits the entry of other sperms into the egg's interior, thus preventing polyspermy. In mammals, fertilization membrane is not formed, but the cortical granules contain enzymes which are proteases, which alter the structure of zona pellucida, preventing polyspermy. Before the onset of fertilization, the process involves the ova will be in hibernating condition with low metabolic rate, and the nucleus is inactive with nuclear division arrested at metaphase stage of meiosis II. With the fusion, the sperm and the ova undergo rapid metabolic changes, which are referred to as metabolic activation. The changes include a change in the metabolic potential, rise in calcium concentration, increase in oxygen consumption, along with increase in protein synthesis, as these are needed for further nuclear divisions. The nucleus of the ova, which remained in the metaphase of meiosis II, becomes active, completing its second division and releases the second polar body. Now, the nucleus of ova is the female pronucleus ready to fuse with the male pronucleus. The egg's nucleus after the first and second meiotic divisions is called as female pronucleus. The compact sperm chromatin is exposed to the egg cytoplasm when sperm nuclear envelope vesiculates upon entry into egg cytoplasm. The condensed sperm chromatin undergoes decondensation by exchanging DNA binding proteins with the egg proteins. The decondensed chromatin acquires a new nuclear membrane to form male pronucleus. The male pronucleus rotates 180 degrees so that the centriole of sperm lies between the sperm pronucleus and egg pronucleus. The microtubules of the centriole of male pronuclei extend and make contact with the female pronucleus, resulting in the facilitation of migration of pronuclei of male and female towards each other. The membrane fusion between male and female pronuclei results in the formation of diploid zygotic nucleus. The fusion of the male pronuclei with female pronuclei is termed as amphimixis. Now the egg is said to be fertilized and the diploidy is restored with the fertilization process, and the zygote is now ready to undergo cleavages to develop into a new embryo. With the restoration of diploidy, with the formation of diploid zygote, marks the completion of fertilization process.